Okay, so good morning, everybody. Welcome to our the last keynote lecture of JOBIM 2021. So as Roderic said, the good thing is that with the virtual conference and the absence of a gala dinner is that we are all on time, awake and ready to listen to the lecture. So today we have the great pleasure to welcome Roderic Guigo. So a quick bio, Roderic has a PhD in statistics. He worked in the field of sequence and genome analysis since the early 90s. So first at the DFCI in Boston, then at the Biomolecular Engineering Research Center, and then at Los Alamos National Laboratory in 1994. He moved back to Barcelona and joined the CRG in 2005, where he coordinates the bioinformatics and genomics program. He is also a professor of bioinformatics at the University of Pompeu Fabra. So his group mainly focuses on the mechanisms underlying RNA production, so basically transcription, as well as post-transcriptional processing like splicing, for instance. So combining high throughput and experimental approaches with computational and statistical methods. So Roderic made tremendous and influential contribution to the field of genomics, also participating in fantastic consortium results we all use as good research parasites, to quote Dan Longo and Jeffrey Drazen in their 2016 paper. So for instance, Roderic worked with the ENCODE consortium to functionally annotate the human genome and to better characterize non-coding transcriptions. He also worked with the Phantom consortium on the same topic. And with the GTEC consortium, he produced fantastic expression data in human tissues and linked genomic variations and gene expression, identifying thousands of eQTLs. So in short, you understood that we all benefit from Roderic work at some point, and it is why I'm very delighted and honored that he accepted our invitation to give a talk at JOBIM entitled The Epigenetic Logic of Gene Expression. So for the questions, I just remind you that we have two options. Either you can raise your hands and we'll open your mic, or you can write your question in the Q&A. Plus, if ever you have, we have no time to answer all the questions, we'll leave the Q&A open after the talk, and Roderic will answer your question in the Q&A directly. So Roderic, thank you from all our community, and I now leave you the floor for your talk. Thanks.
Let me share. I guess that you can see my screen now. Yeah. So, well, I'd really like to thank the organizers, Sophia and Denis, for inviting me to give this talk, and I'm really sorry that I cannot be in Paris with you. I actually would like to thank very much Charles for his introduction. I just want to clarify that within these projects, I had been only a small piece of very large consortiums, and of course the individual contributions are in general minimal. Actually, I believe, I don't know because I cannot see the faces, but I believe that some of the people in my group that contribute a lot to the ENCODE and the GTEx projects, I may actually be sitting or listening to me from their computers, and I'm really, you know, it's Franz, it's Sylvain, it's Thomas, Vincent, and many other people that actually did direct contributions to this project. And today I actually am going to talk about work done mostly by Beatrice, although I will be explaining the work. The work has been mostly done by Beatrice Borsari and Silvia Perez-Luque in my group, and it's the title is The Epigenetic Logic of Gene Activation. And I believe I haven't really given this talk before all this publicly, so that's maybe like the first time that I present this in a large international audience. Anyway, so as you know, there is this idea of the histone code. So the histones, as you know, are the proteins that make the nucleosome, and their tails are modified post-translationally. So the idea of the histone code, or the hypothesis of the histone code, is that these modifications of the histones, mostly in regulatory regions of genes, but have an important role in the regulation of the expression of the genes, right? This is something that has been well known or accepted or in the field for quite a long time. And so the idea is that these are the different H1, H2, H3, H4 are the four histones that make the nucleus and core. And then these are the residues that are modified and the modifications. And as you can see, there are a number of modifications that in principle appear at particular locations around the body of the gene that actually govern the levels of the expression of the gene. And the combinations, essentially the idea within this histone code is that the specific combinations of these histone modifications at the regulatory regions confer to every gene a specialized temporal and spatial pattern of expression. Some of these have a positive effect, some of these have a negative effect, some of them may have a positive or negative effect depending on the circumstances. And as you may know, there has been actually number of papers that or works that actually had shown that indeed there is a strong correlation between the level of some of these histone modifications in the regulatory regions of genes and the level of expression of the genes, and predictive models that can predict expression from the levels of individual or of combinations of the levels of histone modifications have been built with, in principle, high accuracy on the levels of predictions of the expression of the genes. Right. So there's a paper in which actually, by the way, we participated. Some time ago, though, sort of serendipitous, we found that there were genes during developmental Drosophila that were activated without canonically marks of active chromatin, right? So without the marks that you would expect associated to positive gene expression. And I'm sorry to brag a little bit about the cover of the journal where we publish this, because just because this cover is inspired in a painting by Salvador Dali, which has this title that I found as surrealistic as the painting itself. And actually, Dali had made actually quite a few paintings around the time of the discovery of the DNA, about the structure of the DNA. I guess that it looks like he was fascinated by this, by the double helix, and made a few paintings. I'm sorry for this bragging about this. Anyway, we have not been the only ones. Actually, before and after, there have been there have been reports in which the expression of genes have been reported without associated changes in histone modifications. And actually, even though when the association has been reported, there's been controversy in the field whether the histone modifications are the cause of gene expression or actually are the consequence of gene expression. And this is, and I would say that at this point in the field, there is a controversy of what is really the role of histone modifications in gene expression. There are many people that believe that they are very important and they are as or more important than transcription factors in regulating levels of expression of genes. Well, there are other reports, and actually even therapies are actually being explored based on modifying the level of some of the proteins involved in the modifications of the histones in terms to modulate gene expression. On the other hand, there are results in the field that point out that you can see expression without associated changes. So, in an attempt to address this problem, essentially, if you really want to solve the issue of cause of consequence, you, I think, can do essentially only two things: some perturbation experiments in which you alter the level of histone modification, and then you see an effect on the expression of the gene, so that when you perturb a system, you see a consequence, so that you can infer that the alteration, the perturbation that you have done in the system is the cause of the observation. Or you can actually explore, right, the dynamics of the system across time, so that when you see that something happens before something else, right, you cannot really conclude that this something is the consequence of the something else, but it's very difficult to conclude that the something else is the cause of the initial observation, right? So So that's what we did. So we studied the system, which is the induced transdifferentiation from B cells to macrophages. This is a system that was put together by Thomas Raff at the CRG, at the Center for Genomic Regulation. And this is a system in the hematopoietic during hematopoiesis, in which the two branches, right, the lymphoid and the myeloid branch are separated. And this is a forced transdifferentiation from a cell in one of these lineages to the other lineages. And this is induced by overexpression of CBP alpha. And what we did is these cells, special type of cells called glial cells, we actually induce the transdifferentiation and monitor a number of omics variables during 12 time points. This is about, I think it's 7 days. 7 days. And we did at that time, actually we did a number of assays. So we measured the total RNA. We measured also both the cytosolic and the nuclear RNA. We did ribosome profiling, we did proteomics, and we actually monitored a number of histone modifications. All these were done at these time points. Just let me defend the use of this system. There are now well, the system actually occurs with large transcriptional changes, right? These are two very different lineages within the hematopoietic lineage, and therefore the transcriptional differences between B cells and macrophages are very large. And so there are other data sets now available through several developmental processes in mouse, maybe in human. But I mean, development is a very different process. But also the thing is that this data is either in whole body or in tissues, and whatever the case, tissues, even if it's just tissues are very heterogeneous systems made of cells of many different types that may actually be having different dynamics during development. Some cells may differentiate it first, some cells may differentiate it later. And actually, when you do bulk analysis of this, it's very difficult, right, to really know which is the precise order of events. Of course, this could be solved in principle through a single cell approach, but there are not really at this point single cell technologies that can profile, right, in a single cell, in individual cell, all these different histone marks and RNA fractions. And in a sense, if you want to think of this, of course, it's not this, but because this process is highly synchronized, it's like if it was we had actually thousands or millions of copies of a single cell, right? Essentially, the idea, well, it's not true, that this, of course, itself has its own different speed of transdifferentiation, but globally, this we believe is a quite homogeneous system. So essentially, that's what we have. I think that this is really what I like this, because we have this. We are monitoring, right, the action of the genome through the cell, from the nucleus, where all the chromatin modifications occur, to the RNA in the nucleus, the exporting to the cytosol, the binding to the ribosome, and the production of the proteins. And we have this through time. So this is a very complex system. And to be honest, the difficulty has been in the analysis of this data, right? I think that we're not intelligent enough to mine this data. I think that this data, we are going to release it soon. I think will be useful for others that may be more equipped to the analysis of the data to actually mine. We have a lot of expectations of the value of this data as a resource, right? Today I will actually focus only on the relationship between the histone marks and the RNA production. So essentially, the idea that how histone modifications relate to gene expression, if any. So essentially, that's in a sense not a very difficult data metric. So for each gene, right, we have 12 measures of gene expression, which are the infer from RNA-seq. And then for each gene, we also have, for each signal, I think there are 9 histone modifications. We have the same thing, right, for 9 histone modifications. And we want to understand the relationship between gene expression and histone marks. And we made some decisions, which are not fully arbitrary, on where to measure the intensity of the histone mark, because of course, as you know, this is done by ChIP-seq, and so there are many caveats in the processing of the data. So we have to call peaks. There are many ways of calling peaks, of measuring the strength of the peaks, etc., etc. So we made some decisions that, of course, can always be discussed with some level of arbitrarity. We think they are well funded, but anyway, so the idea is that we have a signal for each histone mark and each time point. And this is the data matrix stance, and not too complicated in this case, with which we are going to work. And well, nicely, this is a PCA, on which actually this is a PCA of all that, although this is displayed separately of all the data together. That's the reason why the variance captured by the PCA is maybe not very large, but we can see. So that the size of the circles correspond to the time point. The larger the size, the later in the transdifferentiation. We can see that the actual expression recapitulates quite well the transdifferentiation process. We think that this is the repression of the B cell genes, and this is activation of the macrophages, right? This is B cell to macrophages. So this process requires the repression of the genes specific of the B cell type, phenotype, and the activation of the macrophage. And we see that some marks also seem to reproduce well or to recapitulate well the process, and other marks did not. So essentially, we just wanted to something very simple. I can move this thing. I don't know whether I can move this thing here. See, maybe I can move it somewhere else. It's bothering me a little bit. Okay. Yeah. So essentially, what we did, since we have the time points, we have the genes, we actually can try to replicate the work that has been done when computing the correlation between gene expression and histone modifications, right? And indeed, when we did that, we observed more or less what has been reported. So this is the correlation between gene expression and H3K27 acetylation across at each of the 12 time points, and the correlation is quite high, maybe comparable to the correlation that has been reported. And for negative marks, the correlation is negative as expected. But of course, what we have now that I think is not available before is that now we have this data across 12 time points, and what we can do is now actually see what I believe is more relevant is whether changes in gene expression, right, are really across time, are really co-occur with changes in histone modifications, because this is actually what we really could reflect. Could reflect. I'm not saying all know that correlation could reflect some association between histone modifications and gene expression. And what we see is that the expression is lower, right? This is measured. We measure this correlation per gene. This is the distribution. This is the distribution of the values of the correlation. This is the distribution of the correlation between expression and the marks across all genes. And as you can see, the values are lower, right? Substantially lower, and in some cases actually very low, right? For marks that are typically associated to active gene expression, and even in some cases, for instance, in the case of H3K9 trimethylation, which is usually assumed to be a repressive mark, what we see when we look at the correlation across time is actually positive correlation. So we see many genes in which the variation in the levels of H3K9 trimethylation is positively associated with gene expression. So why is this? Why are we seeing actually? I think that this to some extent reflects that the correlations that are computed on a steady state are somehow artifactually bad. And I think that there is a nice demonstration. I don't know what you think. I like this, but many people don't. But we did just the following thing. We just randomized, right, the association between the expression and the levels of the histone modification. So these are the time points, and these are the true associations, right? This is at 0. We have this expression at this level of H3K4 trimethylation. At 3 hours we have this, etc., right? And these are the values of correlation that we have computed. Now we just randomize this. Therefore, there is essentially no association between expression and chromatin modifications. And of course, when we compute the correlations across time, the correlations are 0 as expected, right? Because we have this essentially no association between histone modifications and gene expression. But when we compute this at steady state conditions, the correlation is quite high, even though I emphasize there is no association, there is no biological association between gene expression and histone modification. And this is, I don't know, maybe this is an example or not of the Simpson paradox, I don't know. But anyway, this is something that arises mostly because there is a large number of genes that are silent and they are never marked with histone modifications. And there are a large number of genes that are always highly expressed, never changed in any condition, and they are always marked with chromatin expression. But this doesn't really mean that the particular pattern of variation of gene expression of a gene across time in a developmental or differentiation process, it's really associated with the levels of histone modifications. And I think that here you can see it well, right? This is the expression of the genes. This is, I think, the same in all cases, more or less. And this is the levels of the histone modifications. The red, the stronger the modification and the higher the expression. And what you can see, this is the 12 time points. You see that genes that increase expression, and concomitantly there is an increase on the mark, that decrease expression, and there is decrease of the mark. So there are genes, right, in which indeed, right, there is a correlation, more or less, or an association between the level of the and between the levels of gene expression and the levels of the mark. So the dynamics are consistent. But there are many genes that actually have a similar pattern of variation of gene expression, right, which occurs without any changes in histone modifications. For some marks, typically associated to gene expression, a large number of genes change expression in a very clear way, without association without associated changes in histone modifications. And actually, and the conversely is also true. So these are genes that are stable expressed during transdifferentiation. They don't change expression, and we have genes that do not change expression, but we see changes of histone marks, and genes that are not even expressed during transdifferentiation, in which we also see changes of chromatin modification. So to try to understand how the patterns of histone modifications change through time, we have well, sorry, let me do a conclusion. Let me have some conclusions on the way. So far, I think that the main conclusions of this is the overall association between gene expression and chromatin marking is weaker than previously thought. Many changes in gene expression occur without associated changes in chromatin marking, and vice versa. Many changes in chromatin marking occur without associated changes in gene expression. And actually, this temporal association of gene expression with some histone modifications, most notably with H3 kinase methylations, runs in the opposite direction than previously thought. So to understand, to test this, but still the idea that there is a histone code, so there is a combinatoric effect of the histone marks in the gene expression. We actually to understand the correlated dynamics of chromatin modification across the transdifferentiation process, we employ the macrophage model. This is just, I want to emphasize this because sometimes it's not clear, right? So we have problems with the reference. This is not a typical hidden Markov model like in which you segment the genome based on the combinations of the histone modifications like ChromatinM or Segway and others, right? So this is in this particular case, what we do is to each gene we associate. The gene is defined by the levels of 9 marks, the ones that are we monitoring, right? And then what we are segmenting, right, for each gene, what we are segmenting is the time, right? So we associate at each gene one state, depending, for instance, on the particular. So here we see that the gene is marked by these 3 different histone modifications in green, in yellow, in gray. So let's say that this corresponds to red state. At 3 hours, the gene remains the 3 state. In 6 hours, this peak has disappeared, and then maybe this is the blue state. And it's 168 hours. These 2 marks have disappeared. Only the green mark appears, and this is the green state. So the states define the genes through time. And I actually think this was the original idea on Markov model, right? So I think it's implicit, but in this case, the time is the real time, right? And we are segmenting the time according to the states of the gene, for each gene, we have a segmentation. And what did we find? Well, we found that there were really not many actual combinations of histone marks. So this idea of the histone code, that the combinations of marks define per gene a specific pattern of gene expression across time and across tissues, we didn't see it very supported. Of course, we choose 5 states. It's very difficult with all the states. We run this from 5 to 20, and we choose 5 to some extent arbitrarily, because the things start to, well, with some measures start to flatten out. But anyway, so more or less, this is what we see. Essentially, 5 major types states in which the gene can be. So there are genes in which there is essentially no marking, maybe with the exception of some marking, which is strict and anti-methylation. Then there is genes that are lowly marked. They have marks but are with low levels. Then we have the genes that we call bivalent, because they have strongly marked by negative, by repressing marks like H3K10 trimethylation, and also by H3K9 trimethylation. But they are also marked with active marks like H3K4 dimethylation and monomethylation. And then we have genes that are active, which are marked by all active marks, and then genes that are strongly marked, that are marked by your marks, but actually at a stronger level, right? Yeah. So I think that one thing that also we can see from this categorization of the states is that essentially marking by H3K4 mono and demethylation seems to be a prerequisite by the marking by the rest of the marks. So we don't see states in which the genes are marked by the other marks without H3K4 mono or demethylation. So when we look at for each gene how the states changes through time, so what we see, right, is that most genes actually stay in the same chromatin state through transdifferentiation, right? There are genes that change, right? There are genes that change these states, but many genes, right? Many genes, for instance, these are genes that are always in this active state, or these are in the strongest state, or these are in the bivalent state. So many genes remain in the same chromatin state during transdifferentiation. And even for genes that change a lot gene expression, and this is an example, right? This is a gene, right? That according to our hidden Markov model states it's strongly marked. The gene increases strongly the expression during transdifferentiation, but the levels of the marks remain stable. So the conclusions from this part, there are a limited number of chromatin states. So I mean, this combinatoric thing of histone modifications, I'm not sure is supported by our data. So marking by H3K4 mono and demethylation is a prerequisite for marking by the other histone modifications, and genes tend to remain in the same chromatin state through differentiation, irrespective of the dynamics of gene expression. Still, right, we see genes that change chromatin state, and when we look essentially at the transitions, right, and the transitions between the different states, we see, right, that essentially to get strong marking, you need to go through essentially transitions from lower levels of marking. And what we see is that in the cases in which we see these transitions between states, we do see some association with gene expression, right? This here, the more red means the larger the mean fold change of the genes that go directly from a lower state to a stronger state, for instance, or from a lower state to an active state. So essentially, genes that go from lower marking states to a stronger marking states tend to increase expression, and genes that go from a stronger levels of marking to lower levels of marking decrease gene expression. Okay. So to better understand what is the relationship now between these chromatin states or the chromatin marking and gene expression, well, we did something quite complicated, but because Beatrice's idea I think was good. It's difficult for me to always explain this, but anyway, so essentially the idea is that we look at for each gene, we look at the relation between gene expression and histone modifications, and we determine whether there was correlation, positive correlation, or there was no correlation. and then we cluster, right? We cluster the genes, right, based on the co-occurrence of marks and the relationship of these marks with gene expression. And what we found is that there were essentially three main types of genes, right? So genes in which there was positive correlation with gene expression, about this number. Genes in which, we did this only for genes that were differentially expressed, right? It's only done for genes that are differentially expressed during the process. So there were genes in which changes in gene expression were positively correlated with histone modifications globally. And that's the important thing, is that as we saw with the hidden Markov models, the groups, right, the modifications of the histones tend to occur concomitantly, right? So things tend to change altogether, right? So we see this set of genes that this set of genes, the larger set of genes that they change expression without any correlate changes with histone modifications, which I think correspond to this set of genes here. And then we have genes that are actually gene expression is positively correlated with histone modifications, right? Which I think is this set of genes here. And then genes in which there is changes of gene expression without chromatin marking. And what we saw is that we saw is that among the genes that are positively correlated, essentially were all the genes that were specifically activated during transdifferentiation. So we focus specifically on the following set of genes, because I think those are the genes for which we can actually get more information. So there are genes that were already activated when the transdifferentiation process starts, and there are genes that were activated specifically during the transdifferentiation process. We think that these are very important genes for our understanding, because we know exactly the time point in which the gene is activated. And therefore, right, we could actually see whether we could delineate the order of events that occur before and after activation of gene expression. And what we actually saw is that the genes that were specifically activated during transdifferentiation had a particular strong correlation with gene expression, right? And as you can see, the correlation is very high, and this is one example. And we see, right, increase the levels of the marks during transdifferentiation marks or some others. And if we actually look at genes, these are genes that are upregulated, right? Are upregulated because we look at the genes that were specifically activated during transdifferentiation. We ignore the genes that were activated and then they decrease expression. As a control, we look at the genes that were already activated at the beginning of the process, but were still upregulated during the process. We saw that the correlation with gene expression was much lower. Actually, this is a gene that, as you can see, has a huge increase in expression, in absolute terms larger than the increase in this gene. But essentially, this change in expression is not accompanied by changes in the levels of the histone modifications. So, but we look, for instance, at the degree of upregulation of the different marks through transdifferentiation, we do see a particular order, right, in which we see H3K4 mono-demethylation being activated. This is the percent activation over the 100% at the end of the process, I believe, or something like that. So we see that it should be 100 at the end. I don't know what it is. Something like that. Anyway, so what you can see is that there's a clear pattern, right, in which you see first the activation of these two marks, and expression, and then these marks more or less following gene expression. And when you look at the genes that are already expressed at 0 hours and are also upregulated during the process, like you see here, right, this pattern and this pattern are very similar. Patterns that are very similar. You see essentially very little changes, very little associated changes of chromatin marks. So we try to, as I said, we try to establish some sort of dynamics during this process. Try to some sort of, if you want, what we call this epigenetic logic, right? What occurs before, during, and after expression. And as you can see, these are the 9 time points. These are the different marks. And you can see that there are some marks that have dynamics, that change. These are the genes that are specifically activated during transdifferentiation compared to the genes that are already activated, in which you see very little dynamics. One thing that we realize is that there are essentially 2 types of genes. Genes that were, these genes never get H3 K27 trimethylation, and these are genes that had already H3 K27 trimethylation when the process starts. So if we focus on the genes without H3 K27 trimethylation, we see more or less the following dynamic network of elements. Essentially, the genes go. Essentially, the first marks that they get is H3K mono or demethylation, but this precedes expression. Then, with expression, we see. After that, we see gene expression that tends to occur where H3K27 acetylation and H3K9 acetylation. And after expression, we see marking by H3K4 trimethylation, H3K36 trimethylation. And at the end, we see marking by H4K20 monomethylation. So if we focus, so this is, let's say, we think that this is quite clear. I think it's also consistent with data that has been previously published. For instance, that remarking by H3K4 monomethylation and demethylation has already been reported. When we look at the genes that are marked with H3K27 trimethylation, things are much simpler because these genes are essentially marked with everything before expression. So all marking in these cases for these genes that are pre-marked with H3K27 trimethylation, all active marking occurs before expression, and it's only H3K36 trimethylation which is deposited after gene expression. So how general this thing is? So we look at another model. Again, this is a developmental model, so I think that the data here is less clear, but it is because this is a very complex system. As I said, this is midbrain is only a tissue, but of course there are multiple cell lines, and they may go different speeds of transdifferentiation or differentiation, and we still see that the genes that specifically are specifically activated during development had a much higher correlation with histone modifications than the genes that are already expressed when this developmental process was starting to be monitored. And one thing that we realize is that, in contrast to the human model during development, many genes were pre-marked by H3 by H3K27 trimethylation. So the conclusion, so the global conclusions of this, I think that there is essentially what we see is that there is a strong association between histone modification and gene expression only at the time of initial activation. Further changes in gene expression have been stronger than those occurring in gene activation occur without associated changes in histone modifications. I mean, this may be just what you would expect, right? Essentially, we don't have any demonstration that this is the case, but this could just simply be the saturation, right? Once the modifications have occurred in all the histones, they cannot be further modified. Therefore, once the saturation has been reached, it's impossible. The gene has to may increase expression, but it's impossible to further increase the marking of the histones. And at the time of gene activation, there is a precise maybe quoted order of the position of histone marks with H3K4 mono and demethylation preceding gene activation, H3K27 and H3K9 acetylation occurring with gene expression, and H3K4 trimethylation, K36 trimethylation, and K20 monomethylation following gene activation. So a preprint of this work is available on bioRxiv. We will release the data very soon. And I'd really like to acknowledge the people working on this, but mostly Silvia Perez, Luke, and Beatriz Morsari. Many of these ideas that I presented in this work are their ideas, and I did the best to explain her work to you. Well, thanks a lot for your attention, and I'm happy to take questions if any.
Thank you very much, Roderic. It was really a mind blower. So we do have questions in the Q&R. So I guess you want me to read those questions. So the first one is from Olivier, or from Violaine, sorry. Could you associate changes in histone marks upon gene activations, with splicing isoforms?
Actually, that's a good question. This is a topic that we have explored in the past, but we haven't really looked at the data yet. So I think that this is something that the data should allow to look at, because actually the RNA-seq is quite deep. So we sequence it quite deep. I think that we will have good quantifications of the isoform. So I think that this is really a data a problem that could be addressed mining the data that we have produced, but we haven't looked at that. And it looks maybe odd, but the thing is that it's not so easy to analyze multiple things across time. I mean, it's difficult sometimes to, but I think the data is there and should be used for this program as well.
Okay, so another question from, it's moving so fast, Christelle. So the specificity of many antibodies against transcriptionally active marks has been questioned in several studies. Could it be responsible for the variability found in marks transcript correlation studies? Replication and transcriptions are highly correlated at the level of gene promoters, and differentiation requires the passage through replications to be effective. Have you looked at what marks might be linked to this passage?
Yeah, so for the first question, really, this should be really answered by Sylvia and by Sylvia in particular, who led all the experimental work. I think that we expect, I mean, of course, this could be the cause. So I think that this would affect the comparison between marks, right? But I think that the dynamics of a given mark through time, whether the antibody captures better the level of marking or not, would not affect the changes that we see, right? The changes that we see. Of course, it is, and I agree, it is difficult to make comparisons between the different marks, but I think that within a mark, right, we could actually make comparisons across time. So in this regard, I think that what we are seeing about the dynamics should be robust to these things. And these antibodies that we are using, I cannot tell. I think that they are quite. So I think that the data that we get when we do the controls looks quite good. But what I can do is I will copy some of these questions and actually redirect it to the people in the group that can respond them better. This is one of them. So now you see replication and transcription are highly replicated at the level of gene promoters, and differentiation requires the passage through replication to be effective. I will look at what marks we bring to this passage. And I think that this may be also related to the next question. Can you evaluate the role of cell cycle in the dynamics of different marks? Yes. So for this, that's a good question again, that we haven't looked at the data. So for this process to happen, so cell cycle need to be arrested so that then cells can differentiate it to B cells to macrophages. So there is which actually CBP alpha does, it represses the expression of MYC, which is one of the genes that's involved in the arresting of the cell cycle. And so therefore, yes, exactly. So when MYC is repressed, there is an arrest of the cell cycle, I believe, and then transdifferentiation to macrophages can occur. We haven't looked into this in detail, but I think again, data could be used for looking at this data. We are actually quite interested in one problem, which I haven't commented this, because we have data as well on mouse, right? And on mouse, the process occurs in 3 days. In human, occurs in 7 days. And it's essentially the same molecular process. And we're actually interested in knowing why, right? Why the process is slower in mouse, in human than in mouse. And we think that this is related to the ineffective repression of MYC in human compared to mouse. And I think that it would be really interesting. I agree. What is the role of histone modifications in this project? I'm not sure I answered your question, but I did my best.
Okay, so we have a live question now from Anthony.
Yeah, can you hear me?
Sure, yeah.
Yes, thank you very much, Roderic, for a really, really nice talk, really fantastic data. I have a question related to so the difference that you observe between the genes that show this correlation with the marks and those that don't, because I think that one thing that you're missing here is basically the information coming from the enhancers. Right, it's like also when you look at open chromatin region, we see that at promoters, for most of the promoters, it never changes, right? It's always open and so on. But what really makes the cell type specificity or the response to any stimuli is mainly driven by enhancers. So then I think that really what's missing there is really the relationship of the histone modification marks at enhancers as well. And I think that would potentially help a lot in trying to address the problem of the genes for which you don't find this correlation. What do you think about that?
Yeah, no, I think that you are totally right. And actually the referees also considered that this was important. So we submit the paper. So we actually decided not to resubmit the paper until we did really a work on the answers. The thing is that, I mean, that's easy to say, but it's not so easy to do. Because there are many now ways in which you could connect genes to enhancers, but it's not very clear, right? How to do that. We're struggling with this. We are using high-C data. We are using maybe the ABC data set, whatever. Many data sets that have been created. We have actually linking ourselves, trying to link these correlations between the histone modifications. But then, if you try to use already correlations between histone modifications or between expression and to call the enhancers, you are already in a circular. We are working on this. Actually, the results that we have are too preliminary to present today. One thing that I would have expected maybe, and I can tell you this is not the case, is well, we see that there is this strong association with the promoters at the start of activation, right? Very high correlation. And then we think that the saturation of the modifications in the promoters. Maybe what happens is that later are the enhancers that are actually take over the regulation of the genes, right? The modification of the enhancers. At least this, so far, we have not seen this, right? We have not seen this. We need to look into the data with more detail. That's what we are actually, this is what our efforts are now on this. But again, as I said, this is easy to say. With the promoters, whatever, are very easy to locate. You may be wrong, but you know, and you do this for all the genes in a consistent way, 2,000 bases or 100 or whatever upstream for the transcription site, which more or less well annotated. But for the enhancers, it is difficult. But we, of course, I think that having this sort of logic, right, in which you have this network of events, maybe, as you said, and that's, I hope, although I don't think this will happen, by including the events happening on the enhancers, you may have a better picture of all the events that regulate of the contribution, right, of histone modifications to the regulation of gene expression. And I agree, that's what we're working on now.
Yeah, just one more thing. I think that it's not only when the gene is already activated that then the enhancers might come in. I believe that the enhancers are involved also in the activation. If you look at the Phantom 5 data, they've shown that basically you have activation of the enhancers even before having activation of the gene that they are activating. So I think it's driven mainly by this as well.
Hey.
But, yeah.
No, I agree. You said that maybe one thing that is one of the possibilities that we thought, not the only, right, is that maybe we don't see this correlation because things are already saturated, and then, but I think that something that we could just investigate with this data again is the crosstalk of the modifications between enhancers and promoters and how this changes over time. So I think that this is interesting, and that's what we are actually looking at now.
Great, thank you very much, Roderic. Fantastic, thanks.
Thanks. So questions from Denis now. So your temporal schemes hint to some kind of asynchronous dynamics, potentially with alternative dynamical path across different chromatin states. Are you attempting to build a dynamic model?
Can you just? I was trying to read this, but I didn't see it on the
Maybe
and then I got distracted.
Could you ask the question directly?
Yeah.
Raise your hand.
I see. Now I see. Now this is by the knee, right? Your temporal schemes into some kind of asynchronous dynamics, usually with alternative dynamic paths across different chromatin states. Are you thinking to build? I don't understand what you mean a dynamical model, but we are trying to build a model in which we try to establish, right, which is the order of events that are occurring at the time of gene activation. That's what we're trying to do. And what we see, I don't know whether at different chromatin states, but what we seem to see is that indeed, depending on some of the states, this path is different. For instance, if the genes are remarked with H3K27 trimethylation, the order of events is different if the genes are not remarked by H3K27 trimethylation. Exactly. So there may be there is an alternative order of events. I don't think that there is a single order of events. This was what our initial assumption, and I think this was wrong. I think that there may be it's not a linear network. It's actually a network with different paths. We hope that still there are some dominated paths. Otherwise, if everything was connected with everything in all directions, there will be no association between histone marks, and we think that there is some structure in this network.
So a question from Serra now. In the first slides, there is a violin plot together with dots at each time point. Is it correct that the violin plot represents correlation between changes in expression and changes in histone marking? Is it, in this case, is it between each pair of consecutive time points? From Serra.
Yes, so Serra, nice to hear it from you. Thanks for your question. Yes, exactly. There's a violin plot together with dots at each time point. The violin plot represents the correlation between changes in gene expression and changes in histone marking during transdifferentiation per gene, right? So the dots is the correlation across all genes, right? At a particular time point between the expression of the gene A, gene B, gene C, gene D, and the marking of gene A, B, C, and D, right? This is a typical thing. The correlation at steady state is over 20,000 genes, right? And these are the dots. And then the violin plots, we compute the correlation per gene across time. So we have only 12 time points here instead of 20,000, right? We have the expression of the gene A and the mark of the gene A at time 1, the expression of gene A and the mark of gene A at time 2, etc., etc. And this correlation is much lower than the correlation that would serve at the steady state, which I think I can convince you is partially artifactual. High correlations that have been reported are partially artifactual. No, I think I froze. Oh.
Hello?
Hello?
Yeah, okay. So yeah, it seems I got an internet connection. So a question from François.
Sorry, I was disconnected for a while, sorry. Yeah. Yeah, go ahead.
I think we are losing Charles.
Hello?
No? Yes.
Okay. Yeah. Could you hear me?
Yes, we can hear you now.
Sorry about that. So I was saying a question from François. Are some of the studied genes involved in marking the histones?
Yeah, that's a good question. Yeah. Some of the genes involved in the modifications of the histones, right? The methyl transferases and all these things. I guess that we haven't looked, or maybe we have looked into this, but I don't know the answer. But I think it would be actually interesting to look at So the problem, we were trying to do some perturbation experiments by knocking down some of these enzymes, but it's very difficult to do this. So maybe looking at the expression could be an idea. Yeah, we haven't.
Okay. Question from Claudia. Have you checked the variability of the different marks across genes and time as function of the expression changes? Can you comment?
Yeah, maybe that's something that we should look, right? You mean about whether, I guess if I understand, is whether the variance of the marking by a particular histone modification is associated to the level of expression. So genes that express higher levels have more variance or less variance. We haven't looked at that. Maybe we should. Oh, thanks for the questions, actually. That's a lot of work that you are giving us. I should have invited this Beatrice and Sylvia here for your great suggestions. Okay.
Okay, so maybe one last question from Sylvain. What is the next scientific controversy you are planning to investigate?
Well, hi Sylvain, how are you? Well, I think that I'll focus on the party next week. I'm the controversy, but I hope I'll see you in a week from now. No, I don't know. As you know, yeah, I like to investigate things. I know I'm not very familiar with this. I mean, this is not my area of expertise, right? I'm not a person working on epigenetics, but I found this idea of the relationship between histone modification and gene expression sort of an interesting problem because it is unclear. And because it is unclear, even if you don't know a lot about the field, I think that you can make contributions, and that's the reason when we work on this. Anyway, thanks for the question.
Okay, so I think we should move on now. So thanks again, Roderic, for this super nice talk.
It's very
bravo. Okay, so thank you.